All right, welcome everybody. You are the dedicated, the smartest of all the people that attended the World Conference on Lung Cancer, staying through the bitter end. I'm Melissa Johnson, and I'm thrilled to welcome you to taking aim at the next wave of antibody-drug conjugates in non-small cell lung cancer. We're going to focus today on two of the antibody-drug conjugates that are rapidly making their way to approval in your country, hopefully, soon. We're going to focus on TROP2 and HER3. Let's see. But without further ado, I want to introduce my colleagues who are going to take us through a lot of the data today. There are three colleagues I want to introduce you to. First, Professor Hide Hayashi. He's a professor in the Department of Medical Oncology at Kindai University in Osaka, Japan. I also want to introduce you to Dr. Antonio Passaro, who works in the Division of Thoracic Oncology at the European Institute of Oncology in Milan, Italy. And last, but certainly not least, Dr. Enriqueta Felip, who's the section chief, the head of thoracic oncology, and a professor of medicine at Valdebrán in Barcelona, Spain. Also at the University Central de Catalunya. So we got all corners of the globe represented today, and we hope that you enjoy today's presentation. Let's see. You know, you can go to peerview.com, 23ADC, to look up the data that we're presenting today. You can also download resources. Some of the slides we have are fantastic, but they're going to be small up here on this big screen, and so you can learn even more by going to this website after today's presentation. Okay, so here are our three goals for today. We're going to improve your knowledge of the composition, mechanisms of action, rationale for use, and clinical potential of novel ADCs targeting TROP2, HER3, and we'll have a brief mention of others right at the end. We want to improve your awareness of the ongoing clinical trials, evaluating these drugs so that when the trials have efficacy data to share, you'll understand where they fit into your clinical paradigm. And we want to improve your skills in integrating these antibody-drug conjugates into your clinical practice for treatment of non-small cell lung cancer patients. Lofty goal. Okay, so let's jump right in. You know, these days when we talk to our lung cancer patients, we talk to them when they're newly diagnosed about targeted therapy, we talk about immune therapy, and we talk about chemotherapy. And all patients, hopefully in your country too, will receive some combination of those therapies. But of course, the truth is that resistance inevitably develops in our patients. We're not yet curing very many people with lung cancer. And so we need to have other options for these patients beyond these frontline standards. So today we're going to discuss how antibody-drug conjugates coming along could join the treatment paradigm for these patients. You'll see this again at the end, but essentially this is one of those slides that you might need to look at a little bit more closely later. But this is evaluating sort of the biomarkers that we use, PD-L1 and NGS, for biomarkers like EGFR and HER2 to help assign therapies for our patients in the front line and the second line. And so this is we're going to put on this paradigm by the end of the hour, the new ADCs. So if you're coming to this session today because antibody-drug conjugates are not something available to you in your practice, they took you by surprise at this meeting and you thought, before I go home, I need to make sure that I understand them, you're not alone. This slide details data from a longevity survey that was performed at ASCO this year. On the left-hand side is the data from doctors, and on the right-hand side is, the other way around. On the left-hand side is data from patients that were surveyed, and on the right-hand side, data from doctors surveyed about antibody-drug conjugates at that meeting. And what's remarkable is that the majority of patients were not at all or only a little bit familiar with these antibody-drug conjugates. And likewise, doctors vaguely understood what they are, but were unsure about who the patients were in lung cancer where these drugs would apply. So you're among friends here. And so let's just jump right into the data. We're going to get to know antibody-drug conjugates. We'll start with a polling question. In order to access this polling question, you have to scan the QR code. So please do that now. Which of the following is not a main structural component of TROP2 and HER3 targeted ADCs in development? Number 1, the TROP2 or HER3 directed antibody. Number 2, target antigen. Number 3, stable linker. Number 4, cytotoxic payload. Number 5, all of them. Number 6, I'm not sure. Enter your answer now. It's okay to be honest. No one's grading you here. Okay, please enter your answers. They won't stop the music until you do. Okay. All right, we'll come back to that question after this session. Let's see. Okay, let's jump right in then with a discussion of antibody-drug conjugates. Dr. Passaro, can you explain how you think about antibody-drug conjugates? Are they targeted therapy or are they systemic therapy, systemic chemotherapy?
Thank you, Melissa, for the introduction. I think this is the main question when we discuss about ADC. So ADC is a new class of cancer therapeutics. It's an oral target agents are known as standard chemotherapy, but it's a very complex structure in which we can see this a very beautiful cartoon of a review published on lung cancer by Professor Naidu and colleagues that we have 3 main components. So the first is the antibody that is able to interact with the target antigen or cancer cell. The second block is the linker, that is one of the most smart structure that is able to interact between the antibody and the drug. And when we discuss about the drug, we have a different kind of names. Generally, when we discuss about ADC, we discuss about the payload. That the payload is the very simple definition of the cancer drugs in the past of chemotherapy agents. And that these 3 components are the part of the main structure of the antibody-drug conjugates. So in my opinion, it's very important to remind that we are not discussing target agents, also if we use this in some driver-positive disease, but we are discussing very complex moleculars, in which, of course, the use of payload of a different kind of number of payload could be one of the most important key options for the future of understanding of the efficacy.
Thank you. And Hide, Professor Hayashi, will you explain the importance of DAR? What does it stand for, and how does that build on what Dr. Passaro has told us?
Yeah, thank you, Marissa. DAR, drug-antibody ratio, is one of the most important quality parameter of the ADC. And the DAR is presented as the average number of the payload to one antibody. And previously, first approved the ADC TDM1 to approve for the breast cancer. DAR was 3. Meanwhile, the trastuzumab deruxtecan DAR was 8. This difference might be the efficacy difference, I think.
Thank you. And if we go on to the next one, Dr. Felip, can you tell us then how these drugs work?
Yes, thank you, Melissa. So antibody-drug conjugates have a unique mechanism of action. No? These are agents that deliver the cytotoxic molecules to the tumor with high potency and potential less toxicity, and there is also a potential bystander effect that I'm going to comment. So the antibody engagement leads to payload independent antitumor activity with two mechanisms. First, the Fc-mediated stimulation of immune cell. So the Fc region of the monoclonal antibody component from the ADC can lead the ADCC activity, but also disruption of receptor dimerization and also downstream signaling. An important part is the ADC is internalized in the tumor cells. The payload is released from endosomes and lysosomes and takes the effect of cancer cells, leading to cancer cell death. But then there is also the so-called bystander effect. So the payload may enter to the neighboring cells, regardless of the presence of the target, and can also kill these neighboring cells. So different model of action, targeted cell with the delivery of the cytotoxic molecules into the cancer cell, and also the bystander effect.
So with that understanding, let's look at this diagram. This comes from a very nice review article, the first author of which is our own Dr. Passaro, sitting at the front of the stage. And this details some of the antibody drug conjugates, their targets, their payloads, their DAR. Another good resource for you when you look at these materials later. We have one drug, trastuzumab deruxtecan, that has been approved in the United States for the treatment of lung cancer patients. But of course, our breast cancer colleagues have been using trastuzumab deruxtecan as well as sacituzumab govitecan for triple-negative breast cancer, HER2-positive breast cancer, respectively, as well as our bladder colleagues using sacituzumab govitecan in bladder cancer as well. So there is a lot of opportunity to get experience with these drugs. But Antonio, I have a hard time, I struggle a little bit. We are the poster children for personalized therapy. There's no other tumor type where understanding the tumor of the patient in front of you is more important. But yet, is this a biomarker-directed strategy in lung cancer?
No. So in lung cancer, moving from the breast, and of course the first results that was presenting for HER2 targeting 2, we are discussing today new here of biomarker agnostic strategy. And this is very interesting. It's a biomarker agnostic, why? Because we are not targeting a single cancer driver alteration, in particular in our disease, but we are using the different kind of target, and we can see in this picture that generally are overexpressed in the patient with the non-small cell lung cancer, also small cell lung cancer. And this is why there is not the need to target at the present time, of course, based on the previous data that evaluate this expression in lung cancer. And this is why we can use maybe in the future, the use of different kind of ADC, regardless the present or not of driver alteration on a specific target.
Okay, so this is your first test. We're going to go through the polling question one more time. Let's see if, now that you're starting to eat your lunch, if you have the answer now. Which of the following is not a main structural component of the TROP2 and HER3 targeted antibody drug conjugates in the development of lung cancer? Enter your answers again. Maybe your answer will have changed. Number 1, TROP or HER3 directed antibody. Number 2, target antigen. Number 3, stable linker. Number 4, cytotoxic payload. Number 5, all. Number 6, I'm still not sure. Okay, so now let's see how we did here. We got the right answer to this question is number 2, target antigen. And I'm proud of you. You got the right answer. More after the post-test. So great, we'll keep going. And we're going to jump into some specifics, first about TROP2 and then about HER3. Okay. Here's another question. Which of the following would you test to select patients with lung cancer for TROP2 targeting ADC therapy? Would you look for TROP2 expression by IHC? Would you look for TROP2 mutations by NGS? Would you look for TROP2 amplification by FISH or NGS? Would you do no biomarker testing in order to identify patients? Or are you not sure? Enter your answers now. Well, you're accelerated in your development today. Everybody has the correct answer, but we're going to go through the data anyway. Hide, kick us off. Tell us about TROP2.
Yeah, okay, Melissa. TROP2, well known as thromboblast cell surface protein 2, is a transmembrane calcium transducer 2. And TROP2 extracellular domain can form dimers, but their effect of dimerization is not well studied. TROP2 is highly expressed in non-small cell lung cancer and other types of solid cancer. 64% of adenocarcinoma and 75% squamous cell carcinoma. And the high TROP2 expression is associated with poor prognosis and making it promising therapeutic target, I think. Currently, there is no testing recommendation for TROP2, but we can identify by immunohistochemistry.
And Antonio, can you introduce us to the to our two characters here? What are our two antibody drug conjugates targeting TROP2?
So I think that this is one of the most interesting slides of the deck. Why? So we have discussed the same class of agents for the same target for very, very similar trial design that will be presented later. But these are two different moleculars, two different cancer therapeutics. So why? We have a very similar antibody. We have a similar payload, topoisomerase 1, but we have a different DAR. We have a different bystander effect from the drugs, and also we have a different timing of release of the payload. So when we discuss about ADC, in particular TROP2, in my opinion, we do not to homogenize our content, our understanding on different kinds of drugs. And here we can see that we have the two main TROP2 under development at the present time, sacituzumab and dapotopam deruxtecan, that are very, very promising. And focus, my opinion, one of the main characteristics with a very different DAR. In the dato, we have a DAR of 4.1, and for sacituzumab, we have a DAR of 8.1. But it is only one of the differential characteristics of these two kinds of drugs.
This is another great resource in your, that you can access online, hard to see from the back of the room, no doubt. It is a big overview of the ongoing trials using these two agents in lung cancer. So I'd encourage you to look at it because it gives you the design, it gives you the setting, and as well as the estimated enrollment. This is a huge area where we're figuring out the answer. Enriqueta, will you take us through TROPION Pan-Tumor 1?
Yes, thank you, Melissa. So the TROPION Pan-Tumor 01 is a study including patients with relapsed refractory metastatic non-small cell lung cancer. The patients were unselected for TROP2 expression, and also patients with stable treated brain metastases were allowed to be included. There is a dose escalation, and the maximum tolerated dose established was 8 milligrams every 3 weeks. And then there are 3 cohorts of expansion in patients with non-small cell lung cancer. 50 patients were included at a dose of 4 milligrams per kilo every 3 weeks. 50 patients were included at 6 milligrams per kilo every 3 weeks, and 80 patients were included at 8 milligrams per kilo every 3 weeks. So there are other cohorts with patients with triple-negative cancer. So you can see there is also in this trial a number of patients included with actionable genomic alterations, mainly EGFR mutations, but not only, after treatment with tyrosine kinase inhibitor. And there was a presentation of the activity of Dato-DXd in this group of patients with actionable genomic alterations previously treated. So 34 patients, the response rate was 35%, and I think a very promising duration of response of 9.5 months. In this analysis, there was a clinical activity in patients with EGFR activating mutations, but also in patients with EGFR exon 20 insertions and patients with ALK, among others. You can see also the waterfall plot with the tumor change, the volume, and also there were activity in the 3 different doses that we have mentioned. There is also the presentation of the 3 cohorts in patients with non-small cell lung cancer that I have discussed previously. The recommended dose of Dato-DXd in patients previously treated is 6 milligrams per kilo. 50 patients were included, and I'm going to focus on the activity in this group of patients treated at the recommended dose. So the response rate was 28%. The patient's median duration of response, again, in this scenario, is really promising, a median duration of response of 10.5 months. So the response rate was very similar when we compare with 8 milligrams per kilo and 4 milligrams per kilo. You can see also the waterfall plot with the vast majority of patients with tumor reduction in the measurable lesions, and also the spider plot showing that durable responses with this strategy.
Okay, let's move from efficacy to safety. Antonio, tell us about the safety of this compound in the phase 1 study.
So in this very clear slide, we can see that the Dato was evaluated in 3 different doses, and toxicity was not significant, as we can see here, with a grade 3 more or less than 50%, and generally with the same toxicity that we are able to identify for patients that receive chemotherapy. The most common was nausea, stomatitis, fatigue, vomiting that was generally well managed in the clinical setting. So, and starting from this phase 1 that evaluated the 3 different doses, the subsequent step was to evaluate the first phase 3 trial in this particular setting, that is the TROPION-Lung1 trial. That is a trial that is ready to be presented in the next months. That was presented as a press release that showed positive results about progression-free survival. And the TROPION-Lung1 is the first phase 3 trial that evaluates in the biomarker diagnostic strategy the use of ADC, here datopotamab deruxtecan, compared to docetaxel in patients with a previous treated non-small cell lung cancer without a driver alteration. So I think that this trial will be a part of the history of the development of the ADC in lung cancer, and maybe will be presented very, very soon.
Yeah, this is a highly anticipated study, right? Not just because of the results relative to docetaxel, but this will serve as a good baseline for patients who've been treated with chemo and IO followed by docetaxel, right? What actually happens? Do patients do a little bit better after they've had IO and then have docetaxel? I think this is a very important benchmarking trial for the future. Now, let's switch gears then. If it is positive, then of course we'll move it up into the front line. Hide, tell us about the initial Tropion Lung 02, which was the first phase 1b trial to move Dato into the frontline setting for lung cancer patients.
Yeah, thank you, Melissa. Tropion Lung 02 is the first phase 1 clinical trial about the combination of Dato-DXd and ICI and platinum therapy. This phase 1b study evaluating this combination in advanced non-small cell lung cancer without actionable genomic alteration. Patient was advanced or metastatic non-small cell lung cancer, and in dose escalation phase, less than 2 lines of prior therapy, and the dose expansion cohort, less than 1 line of platinum-based chemotherapy. And consisted of the 6 cohort, and 3 out of 6 cohort evaluated the 4 milligram per kilo Dato-DXd, and another 3 cohort, 6 milligram per kilo Dato-DXd. And all cohort included the pembrolizumab therapy, and 4 out of 6 cohort included the platinum therapy, such as carboplatin and the cisplatin. Sorry. This is treatment efficacy in all enrolled patients. Doublet therapy is shown as blue, and the triplet therapy as shown red. And the confirmed and pending overall response rate was 38% for the doublet patients, and the 49% for triplet patients. Disease control rate was 84% and 87% respectively, and the median duration of response was not reached. Preliminary PFS in all patients, median PFS was doublet of 8.3 months and the triplet of 7.8 months. Next slide, please. Notably, in Tropion Lung 02, efficacy in the first-line setting was evaluated. Overall response rate was 50% of doublet therapy and 57% of triplet therapy. And the disease control rate was more than 19%, and the median duration of response was not reached.
Enriqueta, what about safety here as we move this up into the front line with immunotherapy and potentially carboplatin?
Yes, thank you, Melissa. So in this slide, you can see the safety presented in Tropion Lung 02 during the ASCO meeting. So overall, perhaps the most relevant toxicity is stomatitis, that is present in a similar percentage of patients in the doublet therapy or triplet therapy, but in the majority of cases, grade 1 and 2. Anemia, platelet decrease, diarrhea, and neutrophil count decrease was more frequent in the triplet therapy. Decreased appetite, constipation, vomiting, but as I mentioned, in the majority of cases, grade 1 and 2. The special interest for some adverse events, ILD was seen in 17% of the cases with a doublet combination, but only 3% grade 3 or 4, and 22% with a triplet combination, only 3% grade 3. There are no grade 5 ILD reported in this study. And then there is ocular surface toxicity, again, mainly grade 1 or 2, in 16% of the cases with a doublet, 17 with a triplet combination. But, as I mentioned, no grade 5 adverse events of special interest were reported.
And of course, at this meeting, we have the first report of Tropion Lung 04, part of a session on Sunday. Antonio, give us just a broad strokes. You know, we're still unpacking this data, and what are your thoughts about it?
The Tropion Lung 4 that was presented here in the last days, I think a very interesting trial design. Of course, the data that was presented are preliminary, but here we are understanding, we're trying to understand if this kind of combination could be evaluated in the future trial in patients with non-small cell lung cancer. The initial results evaluate patients also in a treatment-naive setting with the combo of dato plus durvalumab, but with 2 different doses, also with a triplet of dato plus durva and carboplatin. And we can see that the over response rate for the doublet is more or less at 50%, with a disease control rate at 92%. And for the cohort for the quadruplet, we have a disease control rate that's very similar, 92%, with an increasing over response rate. Of course, these results are too preliminary to understand the impact in patient candidates to receive immune checkpoint inhibitor, where of course the efficacy of PFS and OS are more crucial, but this is the view to understand that we can proceed for this kind of combination. Of course, here, for this kind of trial of a combination ADC with immune checkpoint inhibitors of a combo with chemo, the safety is one of the most important issues to evaluate. But in this preliminary evaluation, we can see that no major issue was identified for patients that received the combo, and only 4 cases of ILD was identified, and 3 of these was a grade 1. So I think interesting data to confirm that we can proceed in this way.
One interesting question that came in on the chat, Antonio, I'll direct it to you first. What do we know about expression of TROP2 in the patients that have been treated on trials to date? Is it lower or higher based on TROP2 expression?
Yes. So in the preliminary evaluation that was developed by in the trial with the dato, we saw that the response was independent from TROP2 expression. And the response was very, very similar among patients with a high TROP2 expression and patients with a low TROP2 expression. This is, of course, is also related to the potency of the drugs, the DAR, and for these, in the subsequent trial, was not considered to evaluate for this kind of drugs the expression of TROP2 by immunochemistry.
Thank you. And Enriqueta, I might ask you this question, another question that came in the chat. Is there a reason that immune therapy and ADCs should synergize or work together? What do we know about that?
Yeah, I think this is an important question. Is there any synergy between ADC and immunotherapy? I would recommend also a review from Antonio in JCO that is mentioning this question. Probably there are some evidence, but will depend of the payload, will depend of the factors of each individual ADC. So it's something that I think we need to work more in order to know exactly the best ADCs to be combined with immunotherapy.
Yeah, I agree. That's well said. For the sake of time, and completeness, we want to make sure to mention the other TROP2 ADC that actually we've known about for longer. This was a compound made by Immunomedics, IMU-132, now known as Trodelvy, and being marketed by Gilead. And you see here the similar dose expansion study, looking at 2 different doses of this molecule in a basket of patients, including triple-negative breast and bladder, subsequently leading to their approvals, but also lung cancer. So if we just look at the patients in that initial phase 1 cohort that were treated with with lung cancer, with sacituzumab govitecan, we can see a response rate of 16%. This was data that was reported back in 2017 and was really the only data that we had with this compound until this meeting. What do we see in terms of side effects? Some of my colleagues have mentioned this already. ILD, that is a platform-associated side effect for the Daiichi compounds, has not been associated with this, has been infrequent. We see GI side effects more commonly here, nausea, diarrhea, but notably absent from the list of adverse events on the right-hand side of the slide is the mucositis that is characteristic in Dato-DXd or datopotamab. So Also on Sunday, right next to the Tropion Lung 04 data was the Evoke 2 data. This was a trial also in the front line. It evaluated patients in 2 cohorts based on PD-L1. They received Pembro plus sacituzumab in both cohorts. And again, small data set, a little bit bigger than the AstraZeneca Daiichi data set, about 60 patients here. And the response rates, including patients who were PD-L1 high, response rate of 70%. In PD-L1 low, closer to 44% for patients treated with Pembro and sacituzumab. So, promising and a little different, as Antonio mentioned, and so we await more data here as well. Okay, let's finish out the TROP2 section with this polling question. Which of the following would you test to select patients for TROP2 targeted therapy? Since everybody got it right the first time, we won't have to spend a whole lot of time deciding on our answers, but put in yours now. All right. Okay. Yes, so we've got some converters so that the right answer here is no biomarker testing is yet required for selection of patients to be treated with TROP2. Let's move on to HER3, the target of patritumab deruxtecan. Hide, tell us what we know about about HER3.
Yes, thank you, Melissa. I would like to talk about the HER3. HER3 is also known as the ARB3, which is one of 4 ARB family. And the HER3 is expressed in 83% of non-small cell lung cancer. And the HER3 overexpression is associated with metastatic progression and decreased relapse-free survival in patients with resectable non-small cell lung cancer. Yeah, HER3 alterations are not known to be a mechanism of resistance to EGFR TKIs, but our institution detected the elevation of the hereglin, that is a HER3 ligand, increase after the progressive disease of TKIs. Given those, targeting HER3 may be relevant across multiple EGFR TKI resistance in mechanism.
And then Antonio, tell us a little bit about the HER3-directed antibody-drug conjugate.
So, as discussed by Hide, so HER3 is not a clear resistant mechanism of patient with EGFR disease of resistance, but is a mechanism that is well known, and HER3 is overexpressed in patients with non-small cell lung cancer, in particular patients that progress to cancer treatment. So patritumab deruxtecan is one of the most marked ADC that we are evaluating in the last years for patients with non-small cell lung cancer, in particular in patients with EGFR positive disease that progress to osimertinib, and where we know there is a high clinical need, and of course there is a very competitive field at the present time. And this is a very interesting molecule, it's very, very similar to Dato. So we have an antibody with a very effective payload with a cleavable linker. And I think that in the next slide we'll be able to also discuss some very interesting results that was presented during this meeting.
Okay, let's go to the polling question in this section. In what patient population has patritumab shown promising activity? Number 1, HER3 mutated lung cancer. Number 2, EGFR mutated lung cancer after TKI therapy. Number 3, HER2 mutated non-small cell lung cancer after chemotherapy. All or none? Enter your answers now. Okay, we'll move on. Antonio, tell us just the big picture about this table of ongoing trials.
So in this very simple table, we can see that for the use of patritumab, we jump from the phase 1 to the phase 3. At the present time, we have one of the most important trials, the Athena Line 2, that is a biomarker agnostic use of patritumab deruxtecan in patients that are progressing towards imertinib. And this trial, of course, was developed after the interesting data that was presented also during this meeting for Athena Line 1. And this trial maybe will be close to the enrolling in this year. So the Athena Line 2 and also another strategy, for example, evaluating the Mariposa 2 are two of the most upcoming biomarker agnostic strategy in patients with EGFR-positive disease progressing towards imertinib. And here we'll be able to understand also the impact of the long-term efficacy for PFS and OS.
Hide, tell us about the phase 1 study that led to, that introduced us to patritumab deruxtecan.
Yeah, okay. This slide demonstrated the phase 1 study about the patritumab deruxtecan phase 1 dose escalation and the dose expansion study in patients for the previously treated advanced non-small cell lung cancer. This study was consisted of the multi-cohort and the mainly focused on the EGFR mutated non-small cell lung cancer. Firstly, dose escalation cohort consisted of the 3.2 milligram per kilo to 6.4 milligram per kilo. And the dose expansion cohort mainly focused for the patient with EGFR mutated non-small cell lung cancer efficacy. Primary outcome is the DLTs and the summary of the adverse events. Next slide, please. Recently, updated data is demonstrated in the Japanese Society of Medical Oncology meeting. 102 patients received the patritumab deruxtecan as a 5.6 milligram per kilo, and the subset with prior third-generation EGFR TKIs and the platinum-based chemotherapy treated patient was 78. Overall response rate was 40.2% and 41.0%, and the disease control rate more than 70%. More importantly, PFS, median PFS was 6.4 months for both arms, and the overall survival was 50.8 months and 16.2 months. Next slide, please. Interestingly, patritumab deruxtecan demonstrated activity in patients with across with diverse resistance mechanism about the EGFR TKIs, including the secondary mutation or the bypass pathway, including the ERB pathway or the non-ERB pathway. Next slide, please. And the patritumab deruxtecan was demonstrated the manageable safety profile and the low rate of discontinuation due to adverse events. Associated with treatment discontinuation frequently was about 11% for the 5.6 milligram per kilo, and the 9% was for all doses. More than grade 3 treatment-related adverse event was 74% and 64%, and the associated drug-induced death was not observed. ILD was one of the key adverse events about the deruxtecan-based ADC, and totally 7% in 5.6 milligram per kilo cohort was observed, but more than grade 3 ILD was only one patient, and there was no grade 4, grade 5 ILD patient was not observed. And the types of adverse event was generally myelosuppression and the GI toxicity.
Hide, one question that came in the chat, I thought it was a good one. Can you explain why this is thought to work in patients with EGFR mutated cancers? What makes this target a good one?
Yeah, thank you. Yeah, as you know, the EGFR is one of the ERB family, and generally HER3 and EGFR form the heterodimerization, and naturally we think about the HER3 is one of the acquired resistance mechanisms about the progressive disease of EGFR TKIs for EGFR mutated non-small cell lung cancer.
Thank you. So here's some more data that was reported at this meeting, the HERTHENA Lung 1 trial. I won't belabor this schema, only to say that a couple different strategies for dosing were explored. An up titration in an effort to get rid of nausea was contrasted with a fixed dose. And so, Helena, you reported the results in 225 patients, quite a large data set for a single-arm trial, and she reported 30% for patients, anti-tumor activity for patients. All patients in this trial, I should have said, were EGFR mutated. All patients had progression on a TKI and platinum at a minimum. So these are third-line patients with a 30% response rate, a pretty impressive disease control rate. Look at the disease, the duration of response. Another question that came in the chat is, great, these drugs are potent, but do they have durability? I believe that we're starting to see that that's the case. Some of the frontline TROP2 data is still really immature. We'll have to continue following that. Patients in this trial had a median PFS of 5.5 months, pretty great without progression, as well as median overall survival of a year. And likewise, just as Hide told us from the phase 1, this drug works in patients irrespective of their mechanism of mechanism of resistance to their prior TKI. And so that's very important because it means that it is likely to be a therapy we can use in many of our patients. Enriqueta, tell us about the CNS data that was reported in this trial.
Yeah, one of the questions is, are these ADCs active in CNS? No? So these are big molecules. So I think it's really interesting in the presentation and also during the publication in JCO concomitantly yesterday that there are 30 patients that had brain metastases at baseline without prior radiotherapy. So these are patients untreated for brain metastases and were treated with patritumab deruxtecan, and the response rate was 33%, and also a very good median duration of response of 8.4 months. So there are also in the paper and the presentation and examples of the responses of these brain metastases. There were 7 patients included with measurable lesion and also with a partial response in 2 of the cases. So I think, yeah, we have seen activity in CNS. We have seen also our breast cancer colleagues' activity in CNS metastases with trastuzumab deruxtecan in patients with breast cancer. No? So there is a growing evidence that these agents are also active in presence of CNS metastases. And also perhaps toxicity, to mention, because here more than 200 patients were treated with this agent. And again, the most important toxicities, including nausea, but grade 1, thrombocytopenia, decreased appetite, neutropenia. But again, when we have results in more than 200 patients, in this case, ILD was seen only in 5% of the cases, and in majority grade 1 and 2. So well-tolerated agent in this scenario.
Okay, one last polling question, I think. Let's, in what patient population has patritumab deruxtecan, HER3, DXd been shown promising activity? Is it HER3 mutated cancer, EGFR mutated cancer after TKI, HER2 mutated cancer after chemotherapy, or all of the above? All right, We got some demonstrated some knowledge or some knowledge acquired. The right answer here is patients with EGFR mutated lung cancer after TKI. Although we will perhaps see data in other indications at upcoming meetings. Now, here's this great slide again. I'd encourage you to go back and stare at it. It does show Dato-DXd as well as sacituzumab deruxtecan and patritumab deruxtecan, but it does also demonstrate or illustrate trastuzumab deruxtecan approved in the US for patients with HER2 mutated lung cancer, and ifinatamab, another antibody drug conjugate against B7H3 that showed data in small cell lung cancer at this meeting. But while we hover on this slide, there's a lot of great questions that have come in from this audience. I might just go down the line rapid fire with a few of them. I'll start with Antonio. Is there a washout period before we can use antibody drug conjugates after immunotherapy, for example?
Yeah, this is one of the most important questions now. So we have a limited data to understand this, but of course we need to focus more on the safety to understand this potential sequence, as we can see also in the KRAS area, whether we use a TKI and then immune checkpoint inhibitor. At the present time, we have no safety signals to consider that this is an issue, but of course we need more robust data because we are discussing our preliminary data in this field.
Hide, question for you. How about, should ADCs be used in combination with targeted therapies? Or should they be used sequentially for patients with targeted oncogenes?
Yeah, I think there is no available data about the combination of the ADC and the radiotherapy, I think. But that depends on the radiation side. ADC demonstrated the lung toxicity sometimes. So lung radiation therapy combined with the ADC should be taken care. But there is no data currently.
Here's a question, maybe we'll go to Enriqueta for this one. If we don't yet have biomarkers that predict for response to therapy, how are we going to decide which patient to give which ADC? What factors could we look for in our patients that might predict for benefit?
Yeah. Again, this is an important aspect to discuss. It's so that we have, it's not a biomarker, but those patients with HER2 mutations, in fact, we know that the results with trastuzumab deruxtecan are very good, and you mentioned it's approved by FDA and hope to be approved soon by EMA, no, in patients previously treated. I think these are agents that is a smart chemotherapy, but at the end is a directed chemotherapy in the tumor cells, no? And we have worked with chemotherapy for years without knowing a potential biomarker, no? So I think it's important probably to know, as mentioned, the characteristics of each ADC and also to analyze the results presented in the trials and the site. At present, we have the results of trastuzumab deruxtecan in HER2. We have also the results that we have commented of datopotamab deruxtecan in patients previously treated, and also this scenario of patients with EGFR mutations and acquired resistance. But in the future, why not we can give the sequence of these ADCs in a given patient, no? So yeah, a lot of studies to be done.
Here's another question. Maybe if we have time, I'll ask all three. I'll start with Antonio, though. Look forward 5 or 10 years in the future. Are antibody drug conjugates part of frontline therapy? Are they part of second-line therapy? Where do you think that they ultimately will reside?
So we discussed that we are at the beginning of the era of ADC, but more or less are more 30 years that the ADC was evaluated in cancer therapeutics, but now we have a very different kind of molecular technology. So I'm sure that these kind of agents will be the standard of care for the treatment of non-small cell lung cancer. replacing more or less all the chemotherapy agents, but I think that we need the time to focus to understand better biology and, of course, efficacy. So today we discussed that for this kind of data, we do not need a biomarker, but all the ADC are very, very different. So I think that we need to evaluate one by one and to focus on the real efficacy in this particularly heterogeneous disease as lung cancer today.
Hide, any thoughts?
Yeah, I fully agree with Antonio. And as Dr. Enriqueta says, ADC sequence is very, very interesting strategy, I think. In Japan, trastuzumab deruxtecan is already approved for the patient with a HER2 mutation. And there are many patients receive the trastuzumab deruxtecan. And clinically, I think that we can select the types of ADC according to toxicity profile, because that toxicity profile is differ between the types of ADC, maybe types of the payload or the types of the antibody. For example, sacituzumab govitecan demonstrated diarrhea, high number of diarrhea, whereas deruxtecan-based ADC demonstrated the GI and the lung toxicity. Of course, we must manage the toxicity more and more.
Yeah, thanks. And what about you, Enriqueta? Other ideas?
No, I think I agree with all the comments. Perhaps also we have to learn in the future about potential mechanisms of acquired resistance of these agents, no? And also the potential integration, no? Even in patients with early stage disease, no?
Yeah. Okay, we're going to skip through this polling question because we've only got a few minutes left. And, yes, do the polling question. Yep, okay, just kidding. Okay, here's a good one for you. Our patient with newly diagnosed metastatic non-small cell lung cancer is found to have no actionable genomic alterations based on NGS. PD-L1 level is 70%. She began first-line carboplatin, pemetrexed, and pembrolizumab with a partial response, and continued maintenance pemetrexed and pembrolizumab, but after only 3 months, developed progression of disease in the liver. What do you recommend next for this patient? 1, docetaxel. 2, carbogem. 3, TROP2 ADC. 4, bevacizumab. 5, erlotinib. 6, not sure. Enter your answer. All right. I agree. I would say the right answer here is in the future, if approved, TROP2 ADCs. We've got, here are some questions for the panel, but I think that for the most part, we've covered these questions, you know, maybe with the time remaining, I might ask each of our panelists for some parting thoughts about antibody-drug conjugates, maybe how you could talk to a patient about these therapies, how we might incorporate biomarkers in the treatment of patients with antibody-drug conjugates in the future, and taking into account different patient profiles. Where might you find that you're using these drugs the most? So I'll just go down the line. Sorry, Antonio.
No.
Wait, actually, maybe we'll start with Enriqueta this time, and so that she has the hardest job, and then we'll go this way.
So I agree with the idea to evaluate this patient for a clinical trial. If a phase 3, the control arm will be docetaxel, so 2 arms are in the same situation. But in my opinion, for a patient that relapses very, very fast after chemo IO, I think that a tissue biopsy and a concomitant liquid biopsy could be useful to understand if some specific trial will be the key for this kind of patient. So I agree with prioritize this patient for a clinical trial, potential with the ADC according to the trial, but I will be prioritized for a tissue biopsy, not for this trial, but to understand more about this tissue, the liquid, the heterogeneity of the disease.
はい。
So I agree with the idea to evaluate this patient for a clinical trial, potential with the ADC according to the trial, but I will be prioritized for a tissue biopsy, not for this trial, but to understand more about this tissue, the liquid, the heterogeneity of the disease.
Yeah, first of all, let's see the results about the tropion rank 02, the results next month. But I fully agree with Antonio. And additionally, I think the liquid biopsy is also the option to detect any single biomarker because new technology about, for example, the CTC detecting and the IHC for CTC is under developing. And the clinical factor is also important. Clinical factor means types of histology or the with or without AGA.
Yeah.
No, yes, I think discussing with the patients the potential opportunity for clinical trials is always a good option. In this case, it's important that the patient had a NGS diagnosis and there were no any molecular alteration. But yeah, I think this, for example, in my country, we have still problems to have access to NGS, and it's important to know that the patient has no other actionable molecular alterations. I think we are, yes, we want to see the results of the randomized phase 3 trial, no? Because docetaxel is what we have used, no? But it's true that it's a toxic agent and with a limited outcomes for the patients, no?
Yeah. One last polling question before we let you go, turn over the room. Now we have a patient with EGFR mutated lung cancer, receives osimertinib, followed by platinum chemotherapy, and then develops further disease progression. Repeat biomarker testing doesn't show MET amplification, doesn't show C797S. What do you recommend to this patient? Number 1, docetaxel. Number 2, HER3 targeted ADC. Number 3, TROP2 targeted ADC. Number 4, immune checkpoint inhibitor. Number 5, something else. Enter your answers now. Look at that. Yeah. That is fantastic. Of course, we do have, this is a little bit of a trick question, right? Because patients who have been treated with, who have an EGFR mutation, who've had TKI and platinum, are also eligible for TROP2. It's going to be an area of active investigation which of those you use, but certainly patritumab deruxtecan today is likely the best answer. Okay, and with that, we're going to finish our session. Thank you for staying till the end. Please visit us at peerview.com and complete your post-test survey so we can make this better next time. You can also download slides, practice aids there. And you can watch a replay of this event, should you choose, in the next 24 hours and in coming weeks. So thank you again for joining us. Join me in thanking my panel here for a great job. Okay, and most importantly, safe travels home after the meeting, and we'll see you next time.